Hello friends, comrades, fellow creatures of life. This video will go under a reading of Paleobiology and the fossil record. It will go under the science playlist, given that it is a scientific field. And so we will dive into the key points of paleobiology in regard to the field of biology and paleontology, as well as geology. In vain would be the power of vision if it taught nothing. Thomas Paine once said, quote, "Of what use is it unless it be to teach men something, that their eye is endowed with the power of beholding to an incomprehensible distance, an immensity of worlds revolving in the ocean of space." Thomas Paine, 1794. This is a wonderful book, and this is not my lecture. It's not my personal lecture. The only lectures that are not from me are the ones where I don't introduce a book. But this book is by Michael J. Benton and David A. Harper. It's an introduction to paleobiology and the fossil record, in which there are beautiful, absolutely beautiful diagrams, charts, tables, figures, and the like, representing all of geological time and the biology that has occurred thence. I do here only want to go over the main points of paleobiology as a reference. It is good to, as a practicing psychologist in evolutionary psychology, it is good to have a strong primer, not only in primatology, but also vertebrate evolution as well. So, I will go through each chapter and I will read the key points of each chapter. The key value of paleontology has been to show us the history of life through deep time. Without fossils, this would be largely hidden from us. Paleontology has strong relevance today in understanding our origins, other distant worlds, climate and biodiversity change, the shape and tempo of evolution, and in dating rocks. I formerly spoke with the director of biodiversity at the American Museum of Natural History in Manhattan, New York. Paleontology is a part of the natural sciences, and a key aim is to reconstruct ancient life. Reconstructions of ancient life have been rejected as pure speculation by some, but careful consideration shows that they too are testable hypotheses and can be as scientific as any other attempt to understand the world. Science consists of testing hypotheses, not by limiting itself to absolute certainties like mathematics. Classical and medieval views about fossils were often magical and mystical, and most of the time were completely misunderstood, or not understood at all. Observations in the 16th and 17th centuries showed that fossils were the remains of ancient plants and animals. By the year 1800, many scientists accepted the idea of extinction. By 1830, most geologists accepted that the Earth was very old. By 1840, the major divisions of deep time, the stratigraphic record, had been established by geological fieldwork and the use of fossils. By 1840, it was understood that fossils changed throughout time, and by 1860, about the time that the American Museum of Natural History opened, this had been explained by evolution. Research in paleontology has many facets, including finding new fossils and using quantitative methods to answer questions about paleobiology, paleogeography, macroevolution, the tree of life, and deep time. Stratigraphy. Scientists began to study the order and sequence of geological events during the Renaissance period, when artists rediscovered perspective. As has mentioned before in my Saga of Humanity lecture on the Renaissance and Reformatio. Lithostratigraphy is the establishment of rock units forming the basis for virtually all geological studies. Lithostratigraphy stratigraphic units are displayed on maps and measured sections. Biostratigraphy using zone fossils forms the basis for correlation, and it can now be investigated using a range of quantitative techniques. Chronostratigraphy, global standard stratigraphy, is the division of geological time into workable intervals with reference to type sections in the field. Cyclostratigraphy links rhythmic changes with Milankovitch cycles. Successions can be orbitally tuned using the tools of astrochronology. Isotopes and magnetostratigraphy are providing useful means of correlations, especially when fossils and other data are sparse. Sequence stratigraphy can provide more refined frameworks that can also help understanding biological change. Geochronometry is based on absolute time measured in years before present by a range of modern quantitative techniques. And extraterrestrial stratigraphy is a rapidly developing discipline as more data becomes available from the moon and other planets. A quote by Nicholas Steno, who started the basic rules of stratigraphy, and says that 6 distinct aspects of Tuscany we therefore recognize: 2 when it was fluid, 2 when level and dry, 2 when it was broken, and I has proved this fact concerning Tuscany by inference from many places examined by me. So do I affirm with it reference to the entire earth from the descriptions of different places contributed by different writers. This was Nicholas Steno in 1669. His dissertation concerning a solid body enclosed by process of nature within a solid. And this is taken from the Introduction to Paleobiology and the Fossil Record, 2nd edition, published in 2020 by John Wiley and Sons. The 3rd chapter is on paleogeography and paleoclimates. Paleobiogeography tracks changing biotic provinces through time, providing evidence for continental drift. Latitude and temperature are the key controls on biotic distributions. Understanding the ecology and environment of fossil biotas is an essential part of robust paleobiogeographic analyses. 2 models, dispersal and vicariance, have been used to explain past biotic distributions. Multivariate numerical techniques may be used to analyze paleobiogeographic data. Changes in geography allowed faunas and floras to migrate and major groups to radiate and go extinct. Fossils from mountain belts are significant in constraining the age and origin of tectonic events. Fossil data have also proved estimates for finite strain and thermal maturation. Ordovician rocks and fossils from the Ordovician tropical sea now form the roof of the world on the summit of Everest. We know from whence it came. Paleoclimates can be described using climatically sensitive biotas and sediments together with stable isotopes. Climate has been an important factor in driving evolutionary change at many different levels. Feedback loops between organisms and their environments indicate that the Gaia hypothesis is a useful model for some of geological time, and we are now living in what many describe as the Anthropocene. Humans are having a major effect on the present and future of our planet with regard to paleoclimates. Paleoecology. Fossil organisms provide fundamental evidence of evolution. They also allow the reconstruction of ancient animal and plant communities. Paleoecologists study the lifestyles of single fossil organisms or the composition and structure of fossil communities. The paleoecology of fossil organisms can be described in terms of their life strategies and trophic modes together with their habitats. Virtually all fossil organisms interacted with other fossil organisms and their surrounding environment. Populations in paleo communities may be analyzed with a range of statistical techniques. Evolutionary paleoecology charts the changing structure and composition of paleo communities throughout time. There have been marked changes in the number and membership of Bambachian megaguilds, or groups of organisms with similar adaptive strategies. The depth and height of burrowing and tearing, the intensity of predation, and the composition of fossil concentrations through time. Ecological events can be classified and ranked in importance. They can be decoupled in effect and significance from biodiversification events. And a quote from Sir Isaac Newton shortly before his death in 1727. He says, "I do not know what I may appear to the world, but to myself I seem to have been only a boy playing on the seashore, and diverting myself and now and then finding a smoother pebble or a prettier shell than ordinary, whilst the great ocean of truth lay all undiscovered before me." This is on taphonomy and the quality of the fossil record. Hard tissues of plants and animals are most frequently preserved in the fossil record. Soft tissues usually decay rapidly, but rapid burial, absence of oxygen, and early mineralization may prevent decay in cases of exceptional preservation. Physical and chemical processes may damage hard tissues during transport and compaction. Tough portions of plants may be preserved as pre-mineralized tissues, carbon-rich compressions, or cemented casts. There has been a long-standing debate about the fidelity and quality of the fossil record. The fossil record is clearly incomplete and biased, but it appears to be adequate when studied at particular scales to document aspects of the history of life. There is strong evidence that the correlation of rock and fossil records through time reflects the third factor, primarily sea level, supporting the common cause hypothesis. Quantitative studies suggest that knowledge of the fossil record is improving, and paleontologists can use phylogenetic trees and fossil records, both largely independent of each other, to establish congruence between the two data sets, and so assess how far the fossil record tells the true history of life. This section is on fossil form and function. Variations in form include normal levels of individual variation between members of a species, as well as variation that results from geographic distribution, sexual dimorphism, different males and females, different growth stages, or ecophenotypic variation, or changes in form occurring within the lifetime of an organism as a result of the environment. Fossil species may show allometry, or changes in relative proportions during growth. Specific organs may show positive, which grow faster, or negative, which go slower, allometry. Morphological variety or disparity may be measured and documented in order to explore linkages between biodiversity and disparity. Generally, in evolution, both are decoupled and follow their own trajectories throughout time. The development of an organism may give some evidence about phylogeny. Changes in developmental rates and timing, which is heterochrony, may affect evolution. The new Evo-Devo perspective shows how certain developmental genes control fundamental aspects of form, such as symmetry, front-back orientation, segmentation, and limb form. Inferring function from ancient organisms is difficult. There are various methods of doing this. Comparison with modern analogs, biomechanical testing, and using circumstantial evidence. Modern analogs may provide exact parallels with some fossil organisms, but more often they provide only principles or rules. Biomechanical models may be used to assess how the design of an ancient organism matches the hypothetical forces acting on it. An example of finite element analysis, a standard engineering technique. Biomechanical models of locomotion are easy to produce, but it is important to check that all possible gates have been considered. Circumstantial evidence, such as the enclosing rocks, associated fossils, trace fossils, and close study of the fossils themselves, can add considerable information on fossil function. Many such observations are the result of chance preservation. Charles Darwin, in his On the Origins of Species in 1859, quotes that there is grandeur in this view of life, that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. The next section is on macroevolution and the tree of life. Evolution by natural selection is a core scientific model that was set out by Charles Darwin and has been confirmed again and again by every branch of biology. Creationist attempts to promote their beliefs, such as intelligent design or the flat earth, are not testable and therefore are not science. Fossil species are identified according to their external form. This is termed the morphological species concept. Speciation often occurs by the establishment of a barrier and the isolation of part of a previously interbreeding population. Evolution takes place both within species lineages, phylogenetic gradualism, phyletic gradualism, and at the time of speciation, punctuated equilibrium. The first model is commonest among asexual microorganisms that live in the open oceans, and the latter in sexual organisms that are subject to environmental and geographic barriers. Punctuational evolution, where most changes occur at speciation and are separated by long spans of stasis, where the species evolves but within limits, appears common. Many examples of evolutionary trends, one way changes in a feature or features, are in reality more complex. The idea of progress in evolution, change with improvement in competitive ability, is hard to demonstrate. The evolution of life may be represented by a single branching phylogenetic tree. Cladistics is a method of reconstructing phylogeny based on the identification of shared derived characteristics or homologies. Genomic data provides powerful evidence for reconstructing and dating the tree of life. There are major efforts to generate ever larger phylogenetic trees comprising thousands of species, and DNA has been extracted from fossils such as woolly mammoths, but not from truly ancient fossils. Charles Darwin, also on the Origins of Species in 1859, quotes as saying, "As buds give rise by growth of fresh buds, and these, if vigorous, branch out and overtop on all sides many a feebler branch, so by generation I believe it has been with the great tree of life, which fills with its dead and broken branches the crust of the earth, and covers the surface with its clever branching and beautiful ramifications." Then we go to the next section, which is on biodiversity, extinction, and mass extinction. There may be 5 to 20 million species on Earth today, a level of diversity almost certainly higher than at any time in the past. There is a debate about whether life diversified according to a logistic model, reaching a global equilibrium level, or according to an exponential model in which diversity continues to expand without reaching a global carrying capacity. During mass extinctions, 20 to 90% of species were wiped out. These include a broad range of organisms, and the events appear to have happened rapidly. It is difficult to study mass extinctions in the Precambrian period, but these seem to have been a Neo-Proterozoic event between the Ediacaran and the early Cambrian faunas. The Big Five Phanerozoic mass extinctions occurred at the end Ordovician, the late Devonian, the Permian-Triassic boundary, the end of the Triassic, and the Cretaceous-Paleogene boundary. Of these, the late Devonian and the end Triassic events seem to have lasted some time and involved depressed origination as much as heightened extinction. The Permian-Triassic mass extinction was the largest of all time and was probably caused by a series of earthbound events that began with massive volcanic eruptions leading to acid rain and global anoxia. The Cretaceous-Paleogene mass extinction has been most studied, and it was probably caused by a major comet or asteroid impact on Earth. Smaller-scale extinction events include the loss of large mammals at the end of the Pleistocene, perhaps the result of climate change and human hunting. Recovery from mass extinctions can take a long time. First on the scene may be some unusual disaster taxa that cope well in harsh conditions. They give way to the longer-lived taxa that rebuild normal ecosystems. Extinction is a major concern today, with calculated species lost as high as during the mass extinction of the past. The severity of the current extinction episode is still debated. Will Cuppy, on How to Become Extinct in 1941, says that the dodo never had a chance. He seemed to have been invented for the sole purpose of becoming extinct, and that was all he was good for. The next section is on the origin of life. Life originated by fusion of organic molecules in the first billion years after the formation of Earth. The precursor to living cells may have been self-replicating RNA. A time before life originated is termed RNA world. Photosynthesis by various organisms generated molecular oxygen, O2, and the atmosphere became oxygenated at a low level, 2.4 billion years. Later, oxygen levels increased further around 0.8 to 0.6 billion years ago. The universal tree of life constructed from gene sequencing of modern organisms shows that there are two great domains, eubacteria and archaebacteria, the latter archaebacteria, including eukaryota. Eubacteria and archaebacteria are prokaryotes, and these contrast with the eukaryotes. The earliest fossils are bacteria and rocks up to 3.4 billion years ago, with intriguing older records back to 3.7 billion years ago, including biomarkers or chemical signals and possible stromatolites. Cellular fossils 3.4 billion years ago are generally accepted, and records increased from 2.5 billion years ago. The oldest eukaryote cells with a nucleus and organelles date back to perhaps 1.8 billion years ago. Red algae from 1.2 billion years ago show that sex had originated probably much earlier. They show evidence of mitosis, but also meiosis, which is unique to sexual reproduction. Multicellularity arose many times in prokaryotes and eukaryotes. Some of the oldest microfossils comprise multiple cells, but eukaryotic multicellularity enabled cell specification and large organisms to emerge. The next section is on protists. Micropaleontology is a multidisciplinary science focused on the study of microorganisms or the microscopic parts of large organisms. Prokaryotes or unicellular microbes lacking nuclei and organelles include the carbonate-producing cyanobacteria, the oldest known organisms. Their radiation during the mid-Precambrian promoted an oxygen-rich atmosphere. Protists, unicellular organisms with nuclei, include a large variety of organisms with external protective coverings, tests, and cysts. Fossilized protists include those with organic, calcareous, or silicious skeletons. A functional evolutionary arrangement is adopted here, in which we consider protozoans first. Then two important but poorly understood groups, Acritarchs and Chitinozoans, and then the phytoplankton. For aminifera, single-celled animal-like protozoans contain both benthic and planktonic forms with chitinous agglutinated, but most commonly calcareous tests occurring throughout the Phanerozoic. Radiolarians, animal-like protozoans with silicious tests, plant-like protozoans with cyclic skeletons are important rock formers. Acritarchs and chitinozoans are palynomorphs, most commonly preserved as cysts, especially important at Neoproterozoic and Paleozoic seas. The phytoplankton comprising planktonic eukaryotes has had a long history, but these three major groups diversified massively in the Mesozoic and Cenozoic, changing the nature of food webs in the oceans. Arthur Conan Doyle in 1891, in A Case of Identity, says, quote, "It has long been an axiom of mine that the little things are infinitely the more important," end quote. And there are different kinds of protists, but I couldn't really explain the actual pronunciations of the words. They're so tricky to pronounce. I just didn't pronounce them. We have the origin and expansion of the metazoans. Relatively few basic body plans have appeared in the fossil record. Most animals have a triploblastic architecture with three fundamental body layers. Molecular data shows that there are three main groupings of animals. The deuterosomes, the lophotrochozoans, and the ecdysozoans, plus a few other taxa, together with the lophotrochozoans and ecdysozoans are usually called the protostomes. 5 lines of evidence, body fossils, trace fossils, fossil embryos, the molecular clock, and biomarkers suggest that the metazoans had originated prior to the Ediacaran of 600 million years ago. Snowball Earth, by coincidence or design, was a pivotal event in the Metazoan history. Bilaterians evolved after the Marinoan glaciation period. The first Metazoans were probably similar to the Demosponges, occurring first before the Ediacaran period. The Ediacaran period biota was a soft-bodied assemblage of organisms reaching its pivot during the late Ediacaran, representing the earliest ecosystem dominated by large multicellular organisms. The small shelly fauna was the first skeletonized assemblage of metazoans. This assemblage of early Cambrian microfossils contains a variety of phyla, where shells are mainly composed of phosphatic material. The Cambrian explosion generated a range of new body plans and a completely new ecosystem during a relatively short time interval. The Great Ordovician biodiversification event was marked by accelerations in diversification at the family, genus, and species levels, together with increased complexity in marine communities. The Nekton Revolution filled vacant ecospace and the water column during the late Silurian and Devonian. Charles Darwin says, consequently, if my theory be true, it is indisputable that before the lowest Silurian, Cambrian, or modern usage stratum was depositive, long periods elapsed, as long as probably far longer than the whole interval from the Silurian age to the present day, and that during these vast yet quite unknown periods of time, the world swarmed with living creatures. This is a lot of scientific material. I have to take a break. Alright, now we get to chapter 12, and this is the basal metazoans, sponges and corals. Parazoans are a grade of organization within the metazoans composed of multicellular complexes with few cell types and lacking variation in tissue or organs. Sponges are almost entirely filter-feeding members of the sessile benthos. The group contains a variety of grades of functional organization. Sponge reefs were dominated during the most of the Phanerozoic by calcareous grades developed convergently across the phylum. Silicious sponges were important reef builders, mainly during the Mesozoic. Stromatoporoids are a grade of organization within the porifera within a secondary calcareous skeleton, important in reefs during the mid-Paleozoic and mid-Mesozoic. Archaeocyths are Cambrian organisms of sponge grade. They were mainly solitary, but developed a branching modular growth mode and successfully built reefs in often turbulent and unstable environments. Reef-type structures were already present in the late Precambrian, hosting large, robust colonial organisms. The Cnidarians are the simplest of the higher metazoans, with a radial diploblastic body plan and stinging cells, or cnidoblasts. The phylum includes sea anemones, jellyfish, and hydra, together with the corals. The Paleozoic and tabulate corals displayed a wide range of growth modes, often related to environments. Neither group was a dominant reef builder. The Scarlactinians radiated during the Mesozoic, with zooxanthellate forms dominating biological reefs. Scarlactinian-like morphs and Paleozoic faunas arose several times independently from anemones with Scarlactinian-type polyps. Reef development through time was waxed and waned, dominated at different times by different groups of reef-building organisms. And, okay, well, enough of that. We're going to go to the next chapter. Okay. Lophophorates. 1. Brachiopods and birozoans. 3. Lophotroschozoan invertebrate groups have lophophores, a filamentous feeding organ, brachiopods, birozoans, and phoronids. Brachiopods are twin-valved shellfish with a lophophore and usually a pedicle adapted to a wide range of life strategies on the seafloor. The phylum Brachiopoda is currently divided into 3 subphyla: the Lingoliformians with organophosphatic shells, and the Craniformians and Rhynchronoformians, both with calcareous shells. Paleozoic communities were dominated by orthids and strophominids, together with a variety of spire-bearing forms, typical of lower biodiversity post-Paleozoic brachiopod assemblages. Brachiopods dominated the filter-feeding benthos of the Paleozoic, but never fully recovered in abundance or diversity from losses during the end Permian mass extinction. Living brachiopods are relatively rare, occupying most cryptic and deep-water habitats. Birozoans are colonial invertebrates with lophophores. They commonly display marked non-genetic variation and occur across a wide range of environments. The Stenolemata dominated Paleozoic Birozoan faunas, with only the cyclostomes surviving the combined effect of the end-Permian and the end-Triassic mass extinctions, as the cyclostomes continued to decline after the end-Cretaceous extinction event. The Chilistomes radiated to dominate Cenozoic assemblages. Okay, we're going to go to chapter 14 now. The Lophotroschozans. Oh gosh. 2. This is mollusks and annelids. The phylum Mollusca can be traced back to at least the late Precambrian, when Cambrella probably fed on algae in Ediacaran communities. Early mollusks were characterized by some short-lived unusual forms, but with the molluscan features of a mantle, mineralized shell, and radula. These were members of the small shelly fauna. Mollusk shell shape and even ornament can be modeled by a variety of computer-based software packages. Only a small percentage of theoretical morphospace is occupied by living and fossil mollusks. Bivalves are characterized by a huge variety of shell shapes, dentitions, and muscle scars adapted for a wide range of life strategies in marine and some freshwater environments. Most gastropods undergo torsion in early life. They have a single cell, often coiled. The group adapted to a wide range of environments, from marine to terrestrial. Cephalopods are the most advanced mollusks, with a well-developed head, senses, and a nervous system. They include the nautiloids, ammonoids, and the coleoids. The group is carnivorous. During the Mesozoic, many mollusks developed protective strategies against predation, such as robust armor or deep infaunal life modes. The group may also have relied on multiformity of shape and color to confuse predators, these annelid worms are a possible sister group of the mollusks. Their jaws and scolecodonts are relatively common in Paleozoic fauna. Now we'll get to chapter 15. Ecdysozoa, or arthropods. Ecdysozoans are protozoans that periodically shed their exoskeletons. They include the Onychophora, Tardigrada, and the Arthropoda, together with the Canorrhyncha, Lorifera, Nematoda, and Parapolida. Arthropods such as lobsters, spiders, beetles, and trilobites have a segmented body plan with jointed appendages, including legs. The first major arthropod faunas of the early Cambrian appear bizarre by modern standards, but probably were no more morphologically different to each other than our faunas living today. Trilobites appeared in the early Cambrian and during the Paleozoic, evolved advanced visual systems and enrollment structures while pursuing a variety of benthic and pelagic lifestyles. The largest arthropods were the chelicerates and include the giant Eurypterids that patrolled marine marginal environments during the Silurian and Devonian. Myriapods represented the earliest terrestrial body fossils in the mid-Ordovician, but track rays indicate the Euthycarcinoids moved into land even earlier in the late Cambrian. Insects first appeared during the early Devonian and diversified rapidly. There are probably 10 million species of living insects. Insects had probably already evolved flight in nymphs before the mid-Carboniferous, when giant dragonflies navigated the forests. The crustaceans included many familiar groups such as crabs, lobsters, and shrimps, together with the barnacles and ostracods. These have a rich fossil record. Much of our knowledge of the history of the phylum has come from exceptionally preserved fossils, particularly from the Cambrian Burgess-Sale, Shangjiang, and Cirrus Passet faunas. Here we have a quote, interesting enough, from Francis Bacon. Whence we see spiders, flies, or ants entombed and preserved forever in amber, a more than royal tomb. Okay, so we're going to go from the arthropods to the deuterostomes, the echinoderms, and the hemichordates. Echinoderms today include sea urchins, starfish, and sea cucumbers. They are all equipped with a water vascular system, including 2 feet, a mesodermal skeleton of calcitic palates with a sterum structure, and pentameral symmetry. During the Cambrian radiation, many bizarre forms evolved. The spindle-shaped Helioplacus may be part of the stem group for the entire phylum, but did not survive the Cambrian substrate evolution. Pelmatozoans were mainly fixed echinoderms and include the blastoids, crinoids, and cystoids. The crinoids include 4 main groups: the Disparida, Cladida, including the Flexibilia, Camerata, and Articulata. The echinoids were part of the mobile benthos. During the Mesozoic, irregular groups adapted for burrowing evolved from the more regular forms that characterized the Paleozoic. Asteroids, or starfish, were more important in post-Paleozoic rocks. Their Triassic radiation probably had its root in deep water hangovers after the end-Permian extinction and may have inhibited the re-radiation of some key groups of brachiopods. Carpoids are traditionally classed with the echinoderms, although some have argued that they were ancestral chordates. They are now accepted as stem group echinoderms. Grapholites are hemichordates relating to the living or rhabdoplurids with similarly constructed tubaria and ultrastructure. Dendroids with autothecae and bithecae, together with many stripes, and graptoloids with generally fewer stripes and only one type of theca, are only 2 most common graptolite orders. Graptolites probably pursued benthic dendroids, plantic dendroids, and graptoloids, and automobile graptoloids lifestyles. Graptoloids evolved rapidly and were widespread. The ideal zone fossils in rocks of Ordovician, Silurian, and early Devonian age. Alright, chapter 17. Fishes and basal tetrapods. Vertebrates are characterized by skeletons made from bone. The oldest vertebrates were small, jawless fish-like creatures from the early Cambrian of China. Armored fissures were abundant in Devonian seas and lakes. After the Devonian, the cartilaginous and bony fishes diversified in several bursts. Conodonts commonly occurred as tooth-like elements that are useful in biostratigraphy, as are some other fish teeth and scales. Tetrapods arose during the Devonian from lobe-finned fish ancestors, and fish-eating amphibians diversified in the Carboniferous. These first reptiles were small insect eaters. Synapsids dominated ecosystems on land during the Permian and Triassic. These groups were heavily hit by the end Permian mass extinction event, and diapsid reptiles, most notably the dinosaurs, rose to become the dominant tetrapods through the Mesozoic. Dinosaurs and mammals. After the Permian-Triassic mass extinction event 252 million years ago, diapsid reptiles diversified in the Triassic. Dinosaurs were a hugely largely successful group for 165 million years ago, 165 million years of the Mesozoic. Pterosaurs were key Mesozoic flyers, and the most important marine reptiles were the Pleistosaurs and the Ichthyosaurs. Birds evolved from dinosaurs and radiated particularly during the Tertiary period. The first mammals were small insect eaters of the latest Triassic. The mammals achieved great diversity and abundance only after the extinction of the dinosaurs. Molecular and paleontological evidence shows that modern mammals radiated in the late Jurassic and early Cretaceous, and that some basic splits were geographic, with major clades separated in South America, Africa, Australasia, and the Northern Hemisphere. Humans arose 6 to 8 million years ago, and fossil evidence points to repeated human migrations out of Africa. Fossil plants and fungi. Fungi have a long fossil record, perhaps dating back to the end of the Precambrian. Freshwater green algae and their relatives are close to the origin of green plants. Plants originated on land in the Ordovician and Silurian, a move first enabled by the evolution of water-conducting tissues, waterproof cuticles, and stomata, and durable spores. Various non-seed-bearing plants arose during the Devonian, but tree-like lycopsids, equistopsids, and groups such as ferns became established by the Carboniferous. These formed the great coal forests. Paleontology, the study of fossil pollen and spores, gives remarkable insights into paleo-environments and paleostratigraphy. The gymnosperms, seed-bearing plants, diversified in several phases during the Carboniferous, Permian, and the Mesozoic. The angiosperms, or flowering plants, diversified dramatically during the Cretaceous and owed their success to fully enclosed and protected seeds, flowers, and double fertilization. And then the last chapter is on trace fossils. Trace fossils represent the activities of organisms. Trace fossils may be treated as fossilized behavior or as biogenetic sedimentary structures. Trace fossils include tracks and trails, burrows and borings, fecal pellets and coprolites, root penetration structures, and other kinds of pellets. Trace fossils are named on the basis of shape and ornamentation, not on the basis of the supposed maker, environment, or stratigraphy. One animal may produce many different kinds of trace fossils, and one trace fossil type can be produced by many different kinds of animals. Trace fossils may be produced within a sedimentary layer, or on the surface. Trace fossils may be preserved in the ground and may be seen as molds and casts on the bottoms and tops of beds. Trace fossils may be classified according to the mode of behavior represented: movement, feeding, farming, dwelling, escape, and resting. Certain trace fossil assemblages appear to repeat throughout time and may give clues about the environment of deposition. Trace fossils often occupy particular levels or tiers in the sediment column, and the depth of tiering has apparently increased throughout time. Trace fossils are of limited use in stratigraphy, except in some special cases. And so there you have it. This is the 2020 publication, second edition of Introduction to Paleobiology and the Fossil Record. And like I said before, those were just the key points. And it took me 45 minutes just to cover the key points. That was very dense and full of terminology, which is very difficult to understand. So if you watched this entire thing, thank you. Like I said before, as a historian and as someone who's going into psychology and trying to study the development of the human mind, you just saw 7 million years of human evolution. And of course, beyond that, we have billions and upon billions of years of biological foundations. So it's a lot to go through. It's a lot to understand. But I'm willing to put in the work, and I hope you will too. Thank you.